the calendar year. For our Thoracic Aortic Rounds, I have the pleasure to introduce Dr. Vaibhav Patel, who's an assistant professor of the Department of Physiology and Pharmacology at University of Calgary, also part of the Libin Cardiovascular Institute. Dr. Patel got his master's and PhD at the University of Baroda in India, and then followed that up with a postdoctoral fellowship from the University of Alberta, Department of Medicine, in translational research. He has several research interests, but some of them are exosomes, intercellular communications, and aortic aneurysms, which is very relevant for these talks and for the people involved in these rounds. So with that, I'll pass it off to Dr. Patel. Title of the talk given to me, Mitochondria, Metabolism, and Aortic Aneurysms. Can we repair the aorta before it ruptures? And Dr. Patel, I'll pass it on to you.
Thank you, Scott. I just realized I didn't update my title slide with the new title. Anyway, so today I'm just going to talk about the aorta. I'm not going to talk about the heart failure and cardiac repair project. So first of all, thank you, Scott, for the invitation, and thank you all for attending this very early in the morning. So we're going to talk about mitochondrial dynamics in aortic aneurysm. And this has been focus of our research for last few years. We got into mitochondrial metabolism and how overall the mitochondrial function affects the vascular function, but also dysfunction, remodeling, and the aortic aneurysm development about a few years ago. And we have been investigating this ever since. And I'm very fascinated with what we have discovered. And hopefully I will be able to excite you about that as well. And I'm going to start by apologizing upfront, because you may see a lot of basic research in here. Some of them may be based in blood and immunofluorescence, but again, that's kind of work we do in the lab, and that helps us understand the mechanism in detail, which is somewhat lacking in the field of aortic aneurysm. Let me start by showing a picture of a genius mind who contributed immensely to theoretical physics, and we're not going to talk about theoretical physics, but most of us know Albert Einstein and his famous discovery of E is equal to mc squared. But really, I don't know how many of us knew that he actually died of abdominal aortic aneurysm. So abdominal aortic aneurysm has claimed a lot of lives across the globe, and it is one of the most common cause of death. I believe it is the 13th leading cause of death in US, costing approximately 15,000. Aortic aneurysm is also a significant medical issue and one of the most common cause of cardiovascular mortality within Canada as well. Although aneurysm affects almost entire aorta, they are broadly divided into abdominal and thoracic aneurysms, primarily when they affect the individual corresponding regions of the aorta. Importantly, irrespective of the region, most aortic aneurysms remain asymptomatic and undetected until a rupture, and that is usually severe and in many cases fatal. Unfortunately, our understanding of molecular mechanisms and pathophysiology of aortic aneurysm remains inadequate, and therefore there is currently no standard treatment for aortic aneurysm, and current pharmacological therapy primarily addresses the underlying risk factors and primarily remains ineffective or less effective. Current medical approach involves monitoring of the aortic diameter by MRI or CT, and then performing open repair or the endovascular repair surgery when the inherent risk of this surgery is less than the risk of aortic rupture. I don't need to talk about this aspect to today's audience. So we are very interested in the regional differences of aortic and within aortic aneurysm, and there are various reasons why we got interested into this. But let me start by a few clinical observations that point to the inherent regional difference. Some of them lies with the prevalence of abdominal aneurysm, which are thought to be, or which are known to be higher than the thoracic aneurysms. Additionally, only about 25% of patients with thoracic aneurysms have concomitant abdominal aneurysms. And multisegmental disease is found in only about 10% of total cases, suggesting there may be differential molecular, cellular, and genetic origins of thoracic versus abdominal aneurysms. And there are various reasons that have been proposed that may contribute to this regional heterogeneity, including elastin to collagen ratio. Now, elastin and collagen both are extracellular matrix protein present in the aortic wall. The ratio of elastin to collagen determines the compliance of the vessel, which decreases if we go from thoracic to abdominal region. The vasomesomes are the small vessels that supply the blood to aortic wall itself. We can even consider this as a vascular density within the aortic wall, which also decreases from thoracic to abdominal region. In addition, perivascular adipose tissue composition changes from thoracic to abdominal region, where thoracic aorta perivascular adipose tissue resembles more brown-like adipose tissue, which is full of mitochondria and is also involved in thermogenesis, whereas the adipose tissue surrounding the abdominal region resembles the white adipose tissue, which has very little mitochondria and is also known to harbor a lot of inflammatory immune cells. Most importantly, the origin of the vascular smooth muscle cells, which are the major cells of medial layer of aortic wall, are also known to be different across the different regions. The smooth muscle cells from ascending aorta are known to originate from neural crest. The smooth muscle cells of the descending aorta are known to originate from somites, and the smooth muscle cells of the abdominal regions are known to originate from splanchnic mesoderm. So basically, they have different embryologic origin as well, and clearly some developmental differences are expected between the smooth muscle cells of different regions. Conspicuously, the regional differences between thoracic and abdominal smooth muscle cells have never been studied and remains the area of research interest in our lab. Our research focus primarily lies in understanding the mitochondrial dynamics in cardiovascular disease. Mitochondria, often known as the cellular powerhouse, which are primarily responsible for ATP production, but also metabolism of glucose and fatty acids. They have a unique capability of going through structural changes in response to various cellular needs. And these are termed as fusion, where smaller mitochondria combine into a larger mitochondria, and fission, where a larger mitochondria divides into smaller mitochondria. This particular process collectively is known as mitochondrial dynamics, and this process is very, very important in regulation of mitochondrial quality control, but also mitochondrial function. DRP1 is, or dynamine related protein 1, is a protein primarily involved in mitochondrial fission. In other words, DRP1 activation divides mitochondria into smaller mitochondria, and we often like to also call this a mitochondrial fragmentation if it occurs in excess, and it has some pathological implications, which we will touch upon in subsequent slides. So DRP1 is a prototype or major protein involved in the mitochondrial fission and is also at the center of our research investigations. So as I mentioned, the smooth muscle cells of thoracic and the abdominal region originate from different embryological regions. So we were very interested in understanding if these different smooth muscle cells respond to stimuli differently, and if they have different phenotype that can be understood and studied molecular and cellularly. So we decided to collect these smooth muscle cells from thoracic as well as abdominal region, and then subject them to various stimuli. And one of the stimuli that we use is angiotensin II, the classically known vasoactive peptide. Angiotensin II in mice is known to lead to a lot of hemodynamic, but also molecular cellular changes. One of them is the smooth muscle cell hypertrophy, hyperplasia, as well as smooth muscle cell death, if we look at it temporally. So we use the angiotensin II as the inducer and subjected both the abdominal and thoracic smooth muscle cells to angiotensin II and looked at their mitochondria. So in these particular images, the red color represents the mitochondrial staining, which is performed at mitotracker red, a dye that is internalized by functional mitochondria. So in these images, we can see the mitochondria that can be delineated using an ImageJ plugin called Mina. And using this plugin, we can really find out how long the mitochondria are, how circular the mitochondria are. In other words, we can really look at the mitochondrial length and comment or identify mitochondrial fission and fusion. In other words, mitochondrial dynamics. So when we subjected both the abdominal and thoracic smooth muscle cells to angiotensin II, both of those cells had smaller mitochondria, as we can see in mitochondrial footprint, as well as mean branch length. In other words, the mitochondria was smaller, but their branching was also lesser in response to angiotensin II. But when we treated these cells with MDV1, which is a pharmacological inhibitor of DRP1, some of those effects of angiotensin II were reversed or prevented, suggesting that the angiotensin II induced mitochondrial changes were DRP1 dependent. So we were happy to see some mitochondrial changes in response to angiotensin II, but we were also expecting to see some regional differences between the abdominal and thoracic. But here the phenotype was very similar, and the effect of DRP1 was very similar. So this was, was a little bit disappointing experiment, or was not as exciting as we actually thought. But we still decided to pursue this and understand if there were other phenotypic changes. And one of the observation that was made by a postdoc in the lab was that the abdominal smooth muscle cells in response to angiotensin II were not proliferating as fast as the thoracic smooth muscle cells. So we actually decided to look into the cell proliferation in these cells, and we used one of the classically used method where the cells are labeled with Ki67 antibody. So Ki67 is a protein which is expressed in the dividing cells in the nuclei. So Ki67 is widely used as a marker of cell proliferation because of its very time-sensitive expression. Using this, we performed the Ki67 fluorescent staining using the Ki67 antibody and secondary antibody that was tagged with the fluorescent dye. And we performed the flow cytometric analysis. And what we discovered is that the abdominal smooth muscle cells in response to angiotensin II were not proliferating, or they had decreased proliferation. This could be also because of increased cell death. But on the other hand, the thoracic smooth muscle cells showed increased proliferation. And both of these effects in the abdominal as well as thoracic regions were MDV1 suppressible or DRP1 dependent. So in other words, the DRP1 activation led to cell death in the abdominal region, but the same DRP1 activation led to increased cell proliferation in thoracic. Now this was very, very interesting. So we decided to look more into the abdominal cells and to see if what was the cause of the cell death. And first of all, we performed one of the staining that we routinely use in the lab. It's called the annexin-5 staining, where annexin-5 is a cell membrane protein which is usually not expressed on the cells, but is expressed in response to apoptosis. So we use the annexin-5 as a marker of apoptosis, and using this annexin-5 positivity and flow cytometric analysis, we discovered that these abdominal cells in response to angiotensin II indeed have increased apoptosis, which was again partially blocked by DRP1 inhibition using the MDV1. And to also link this data with the mitochondrial reactive oxygen species generation, we use the MitoSox, another dye that is specific to mitochondrial ROS. We found that the angiotensin II also resulted in increased mitochondrial ROS production, some of which was also blocked by DRP1 inhibition. So with this, we found that the abdominal cells were undergoing apoptosis in response to angiotensin II, and it was ROS dependent. So next, the reactive oxygen species are often known to be associated with the impaired mitochondrial membrane potential, mitochondrial permeability transition pore, and I don't want to throw a lot of technical terms, but these are some of the mitochondrial properties by which we can judge the quality of the mitochondria. And using some of the widely used methods, including calcine AM and cobalt chloride based assessment of mitochondrial permeability transition pore, we check the quality of the membrane of the mitochondria, which was found to be decreased, or if I were to simplify this, the mitochondria were more leaky in response to angiotensin 2, and this effect was blocked with DRP1 inhibition. So this was crystal clear. The abdominal smooth muscle cells had dysfunctional mitochondria, had a lot of ROS generation, which was associated with leaky mitochondria. Now this is very significant because mitochondria harbors a lot of degradative enzymes, including cytochrome C, which are known to induce apoptosis. But when the mitochondria become leaky, they release this cytochrome C and other enzymes, which are known to result in cell apoptosis. So this was very significant in terms of abdominal cells, but none of these was observed in the thoracic smooth muscle cells. So we then came to an interim summary over here, which suggested that we observed a contrasting phenotype in abdominal versus versus thoracic smooth muscle cells. In both, the angiotensin 2 challenge resulted in mitochondrial dysfunction and apoptosis in abdominal SMCs, whereas angiotensin 2 induced activation of DRP1 resulted in increased cell proliferation. Very contrasting phenotypes. Now, we did perform a lot of molecular assays to understand why there was contrasting phenotype between two different regional smooth muscle cells. And one of the experiments that really stood out was the metabolic assessment of these two cells. And we kind of came to a conclusion that these effects may be based on the metabolic differences between these regions. And to understand the metabolic basis of this contrasting phenotype, we performed a CIHR extracellular flux analysis in these cells. So I won't go into too much detail of what the CIHR extracellular flux analysis is, but in short, what we do is we use these cells, we give them a known amount of glucose and fatty acids. In this case, fatty acids. We use the palmitic acid or the palmitate, and then study the capability to metabolize both the glucose and palmitic acid and generate the ATP. So I won't go into too much detail of what we discovered, but I would like to point out that in response to angiotensin 2, the abdominal SMC showed decreased metabolic potential and decreased ATP production, which sort of aligned well with the increased mitochondrial dysfunction observed in the abdominal SMCs. However, in response to angiotensin 2 challenge, this thoracic smooth muscle cell showed increased glycolytic extracellular acidification rate, and I'll explain what that is in a second, but they were associated with unchanged ATP production. So this kind of indicated that there is an uncoupling between the metabolic potential and the ATP production, which suggested that there may be sort of a Warburg effect going on in the cells, particularly in the thoracic smooth muscle cells. Now, what is a Warburg effect? When we look at the metabolism in normal cells, we know glucose is converted to pyruvate. This is biochemistry 101, and I'm sorry to bring this up here, but I just want to briefly indicate or highlight what the Warburg effect is and what the significance of that is. So in normal cells, glucose is usually converted to pyruvate, and it's using the TCA cycle and oxidative phosphorylation that generates ATP and ROS as byproduct. Whereas in the cancer cells, this particular effect, Warburg effect, is very well known to exist in the cancer cells and very well studied in the cancer cells. So in the cancer cells, the glucose oxidation is largely increased, and that gets converted into pyruvate. But instead of going through the TCA cycle, it usually goes through the anaerobic metabolism and generating a lot of ATP. Now, as we can see, the TCA cycle is usually a lot more efficient in producing ATP, but it's also a very slow process. Whereas the anaerobic glycolysis is very, very fast in generating ATP, but it is very less efficient. In other words, ATP generated per molecule of glucose is much lesser. But cancer cells and a lot of different cells prefer this particular anaerobic glycolysis when they need to undergo rapid proliferation. In case of cancer cells, this can include metastasis. So we kind of discovered that these thoracic smooth muscle cells in response to angiotensin II showed cancer cell-like metabolism. We went into a lot of molecular details to understand why these cells may act differently in response to angiotensin II, be it metabolic differences, be it phenotypic differences, be it ROS generation, or the cell proliferation versus apoptosis. We looked into different pathways, and some of the pathways that clicked are included here. Now, these are classical Western blot images suggesting the levels of different proteins assessed with different treatments at particular time points. So we found that the P53 phosphoretinoblastoma protein, or RB, NF-kappa B, these were all largely changed in the thoracic smooth muscle cells, as well as cyclin E1 and cyclin D1 were also largely affected in the thoracic smooth muscle cells, whereas all of these proteins were unaffected in the abdominal smooth muscle cells. So there is clearly a molecular differences in that, and I'll not go too much into detail of roles of all of these proteins, but I'll highlight that P53, cyclin D1 and E1, these are the proteins involved in the regulation of cell proliferation, whereas NF-kappa B is the protein that is primarily involved in the cellular inflammation, smooth muscle cell phenotypic change. We also discovered that a key protein of NF-kappa B, the P65, translocated to the nuclei in response to angiotensin II in thoracic smooth muscle cells, but not in abdominal smooth muscle cells. And this is very significant because the P65 subunit of NF-kappa B is known to induce a lot of transcriptional changes leading to smooth muscle cell phenotypic change. So summarizing this data so far, we discovered that these abdominal smooth muscle cells in response to angiotensin II showed increased apoptosis, and this was dependent on the mitochondrial dysfunction, leaky mitochondria, and ROS production. Whereas the thoracic smooth muscle cells in response to angiotensin II induced DRP1 activation led to increased phenotypic switch and inflammation and increased cell proliferation. These effects were dependent on P53 cyclin D1 E1 pathway as well as NF-kappa B pathway. Now, just to kind of talk a little bit about phenotypic switch. So smooth muscle cells are known to undergo phenotypic switch to more non-contractile and synthetic smooth muscle cells, which are known to induce the production of extracellular matrix and the ECM remodeling, the key hallmark feature observed in the abdominal as well as thoracic smooth muscle cells, and abdominal as well as thoracic aneurysms. So we looked at the phenotypic change in the thoracic SNCs in response to angiotensin II to validate our initial finding. And indeed, the smooth muscle cells in response to angiotensin II showed decreased ECTA2, decreased calponin 1, and myosin heavy chain 11. These are all the contractile. So these are the genes, but these all encode for contractile proteins in the smooth muscle cells. And at the same time, we saw increased collagen expressions as well as increased interleukin 6 and an inflammatory cytokine in the smooth muscle cells. So this validated the smooth muscle cell phenotypic change, particularly in the thoracic smooth muscle cells in response to angiotensin II, which were again dependent on DRP1. Now, this was all done in the cells, in the isolated smooth muscle cells. So we wanted to kind of move forward in the in vivo models of aneurysm, and we used one of the widely used model of the aortic aneurysm in mice. And let me start by saying that there is no ideal model of aortic aneurysm in the mice or any rodents. But this is one of the most widely used model because it kind of replicates a lot of clinical features observed in the patients with aortic aneurysm. So these mice are ApoE knockout. Now, ApoE is a protein involved in the lipoprotein clearance. And because these mice lack ApoE, they cannot metabolize the lipoproteins. So they do end up getting hyperlipidemia. And when we combine those with angiotensin II, and as we know, angiotensin II is known to induce hypertension and a lot of hemodynamic changes. combine the angiotensin 2 plus ApoE knockout, it does result in generation of aortic aneurysm, both abdominal as well as thoracic, within 4 weeks of angiotensin 2 infusion. This can be validated by increased aortic diameter in both thoracic as well as abdominal regions. Now, when we treated these mice with let me go back to this slide. When we treated these mice with MDV1 for the same duration of angiotensin 2, we saw that the aortic diameter, both the thoracic as well as abdominal, was decreased with the MD1 treatment. In other words, inhibition of DRP1 may be a potential therapeutic approach in this preclinical model, and maybe in larger animal models, and maybe in humans. That's in the future. In addition to angiotensin 2 model, we also use another preclinical murine model of elastase. Now, elastase is an enzyme that degrades the elastin in the aortic wall, which is known to induce the ECM remodeling and the dilatation of aorta and aortic aneurysm. Using the elastase, we developed a model of thoracic aortic aneurysm, which was again prevented with the MD1 treatment. So in other words, when we tried the MD1 pharmacological treatment to inhibit the DRP1 using 2 different murine models, both of those showed regression of aortic aneurysm, prevention of aortic aneurysm, both in the males, but also in the females. Now, this was all done using the murine smooth muscle cells and the murine models of aortic aneurysm. We wanted to understand if there is any translational relevance or potential of these findings. So we collaborated with Dr. Paul Fidak, and we used the human aorta with BAV aortopathy. Now, these were the samples which were, and I know BAV aortopathy is known to be very different than the tricuspid aortic aneurysms. But we use this to really understand if these changes in the mitochondrial structure and function persist throughout the different aortopathies and aortic aneurysm or not. And in these, we compared the aneurysmal area with the remote area within the same aorta. And we looked at the mitochondrial structure in these aorta using the TOM20 mitochondrial staining. TOM20 is a mitochondria specific protein, and by staining for the TOM20 using a fluorescent dye, we can localize the mitochondria, and we can also determine the size of the mitochondria. And to make this smooth muscle cell specific, we can also stain them with calponin, which is a smooth muscle cell specific protein. And in these images, we can see the very nicely stained smooth muscle cells, which also shows the mitochondria, which are much larger in the remote area, but very smaller in the aneurysmal area. In other words, we can see the mitochondrial fragmentation or mitochondrial fission going on in the aortic aneurysmal tissue compared to the remote area. Now, this is very similar to what we observed in the mice. However, this is just an observation in BAV aortopathy. We intend to also understand this in various other aortic disease, including BAV, Marfan, LDS, EDS, and tricuspid aneurysms, and so on. Okay, so in summary, we identified different differential mechanisms of DRP1 that promotes the smooth muscle cell apoptosis in the abdominal aorta and smooth muscle cell proliferation and phenotypic switch, as well as inflammation in the thoracic aorta. Now, these 2 mechanisms are both DRP1 dependent, and they both lead to aortic aneurysm in different corresponding segments. We are more interested in understanding what is downstream to DRP1. In other words, we're trying to understand these question mark, or what are some of the proteins involved in the downstream effect of DRP1. If we can find out what is involved differentially in between the thoracic versus abdominal, we can really understand those mechanisms in deeper and come up with these specific targets that can be utilized to develop precision medicine candidates for thoracic versus abdominal aortic aneurysms. In the next 5 to 7 minutes, I just want to quickly brief you about another project that's going on in the lab, and we have been trying to push this, and to be honest, I've been slacking off on this particular project, but This is a very interesting project, and we are trying to take this to a human tissue level in collaboration with various surgeons, including Scott, Kenton, and Randy. So in this particular project, we are interested in exosomes or the extracellular vesicles. Now, just to not to bore you with a lot of details, these exosomes are very tiny particles. They are usually in nanometer size. These particles are secreted from almost all the cells in our body. Now, these particles were thought to be the cell garbage disposal mechanisms. Although these particles were discovered 30 years ago, nobody cared about these particles until about 10 years ago, when a major discovery by Jen Lotwall and group identified mRNAs and microRNAs in these exosome particles. These particles are now known to carry a distinct amount of cargo, including mRNAs, microRNAs, adenosine molecules, tetraspanins, cytoskeletal molecules, even mitochondria, smaller fragments of mitochondria as well. Now, the way these exosomes or extracellular vesicles work is they are actively secreted from the donor cell, and they travel and they are uptaken by the recipient cells or target cells. And within the target cells, they are degraded and they release their cargo. In other words, they do play a key role in intercellular communication, where they take up the message from the donor cell and transfer it to the recipient cells. But also in this process, these exosomes or extracellular vesicles are also released in the blood and interstitial fluid and urine and various different fluids in our body. Now, our idea with this particular project is to understand if we can harbor the unique signature that is present in the extracellular vesicles and compare those to the one present in the blood sample as well as in the tissues harvested from patients with the aortic aneurysm, and compare those to healthy individuals with an intention to come up with some sort of EV cargo, be it microRNA, mRNA, long non-coding RNA, circular RNA, tRNAs, or proteins that can be utilized to understand a unique, distinct signatures of different aneurysms, and also to use these as biomarkers. Now, this is a project which we recently got Catalyst funding from the University of Calgary VP Research Office, and we are trying to push this in collaboration with Scott, Kenton, and Randy, and hopefully we'll soon have something to show on this particular project too. With that, I would like to thank all the collaborators, various trainees in the lab. Dr. Anshul Jadley has been phenomenal in these projects, and none of this was possible without his hard work, working in after hours and also the weekends and so on. Libin Physiological core facility and various other facilities that support our research, and various funding agencies. And thank you all for listening.
Great job, Dr. Patel. You summarized some very complex stuff in a short period of time, and I have to comment on your pathway diagrams that make it a little bit, at least, something I can digest. I am going to start with some questions. If anybody else has some questions, put up your hand or reach out. The first thing I want to ask you, Dr. Patel, if I follow this in the most simplistic form, so DRP1, cell death, abdo, DRP1, cell proliferation, thoracic, but in your mirroring model, both, regardless of that, you were able to prevent aneurysms in both. So you said that you want to go downstream to find more, but my question is, why do you think the difference, which is, I guess, you're trying to investigate, but maybe you found from a clinical perspective, maybe DRP1 is where you want to be, because it seems to, granted, that's only a mirroring model, but if at that point, for whatever way in your pathway, that stops aneurysmal, regardless of the pathway thereafter, wouldn't that be where you want to focus?
No, that's a very good question, Scott. Thank you for that. So let me just step back a little bit on the DRP1 and how it works. So DRP1 is a very essential protein involved in the mitochondrial fission. Now, it has been known to be upregulated and activated in various disease conditions, but it is also involved in the mitochondrial quality control. And the way it works is, whenever you have a dysfunctional mitochondria, that is removed by mitochondrial fission, followed by autophagy, and the whole recycling of that particular mitochondria. And DRP1 is very essential for that, and various other functions. So is it a good idea to block the DRP1? Maybe it has worked in the mouse model, but again, DRP1 is very, very upstream, and blocking the DRP1 may have some unwanted effects, which we may not discover using just the mutant model that we are using at this point. Going to your question on on how the DRP1 prevented both the phenotypes. So now, you may be at better position to comment on that than me and Paul. Paul may be able to comment on that as well. But in my little experience, thoracic smooth muscle cells or thoracic aneurysms are more affected with the whole ECM remodeling and fibrosis and stiffness and so on, whereas the abdominal aneurysms are known to have increased cell death, the smooth muscle cell death. So in other words, the smooth muscle cells do really behave differently, and DRP1 has different effect, and it did block both of these effects. I guess we were lucky that the DRP1 prevented both these phenotypes. But again, yeah, the smooth muscle cells have different fate in response to challenges in both these regions.
Interesting. Daniel, one of our residents, has his hand up. I'll let him pose his question.
Yeah, thanks so much for your talk, Dr. Patel, and congratulations to you and your lab on some really nice work. I have two questions for you that are not very much related, but the first one is, I found your slide on the tissue that you took from Dr. Fedak and the translational potential of those findings quite interesting. Now, I'm assuming that this was ascending aortic tissue that you got. But I guess one of the things that came to mind is you're going to have differences in hemodynamics across the ascending aorta versus the descending versus the abdominal, and you're going to have, I guess, differences in wall stress across the different regions. So my first question to you is, do you think any of these regional hemodynamic differences contributed at all to the differences that you're seeing with your mitochondrial function and dysfunction?
No, that's a very good question, and that I would like to answer that using the same samples that we used in collaboration with Paul Fedak's lab. So these were collected from the BAV aortopathy, and the way they are categorized is based on the wall shear stress they are exposed to. In other words, the hemodynamic differences. So we did discover that the mitochondria was in fact sensitive to the hemodynamic changes from that particular slide. But again, if I look at the murine data that we have been collecting with respect to angiotensin II model, which is known to induce a lot of hemodynamic changes, the DRP1 was activated and induced the mitochondrial fission. But then we also used another model which was sort of not as much hemodynamic changes dependent. That was using the elastase, which is the local application and very acute application of elastase on the thoracic aorta, and that does develop aortic aneurysm, which is primarily driven by ECM remodeling and elastin degradation and so on. Does it have hemodynamic changes? Maybe, but it probably will not be as severe as the angiotensin II. And we still discovered that the DRP1 was activated in those aorta, and MDP1 prevented the whole phenotype. So I think the hemodynamics plays a major role here, but I don't think it's just the hemodynamics which is involved in the regulation of DRP1. And we're trying to understand what are those other molecules or insults that activate the whole DRP1 cascade.
Ali Fatih, one of our other residents, also has his hand up for a question.
Thanks, Dr. McClure. Thanks, Dr. Patel. Great talk. Daniel asked one of my first questions. The other question, I have very interesting work, especially with the P53 pathways. It's fascinating to see there's obviously some commonalities with other cellular processes that happen. Have you guys looked into DNA methylation in that tissue, the tissue that you've collected, any post-translational, like histone modifications or any non-coding RNAs to see if there's any epigenetic modifications with these tissues?
Yeah, no, that's a very good question, Ali. So you kind of read my mind on that. So I was actually attending one of the talk from Kathleen Martin's group, and they have really nailed this down, and they have really correlated the whole epigenetic changes in the smooth muscle cells, primarily driven by methyltransferases, and the smooth muscle cell phenotypic change. And we were really interested in looking into that. I think we are planning to start with kind of comparing the transcriptomics of these cells first, but that remains in the future plan, and that's a very, very important question. There is protein which we found elevated in these the smooth muscle cells particularly is known as EZH2, and it's been known to drive the whole methyltransferase mechanism and epigenetic changes, but we haven't really pursued that. But that's a very interesting topic, and thanks for that question.
Thank you. Thank you very much.
Great, guys. If there are any more questions, Daniel, your hand's still up. Is that just hanging out there, or you got another question?
Well, I have another question, and then
Yeah.
I promise I'll stop. Yeah. Dr. Patel, my other question for you is your I guess in vitro work focused quite a bit on the smooth muscle cells, which is sensible, like with, as you say, its role in extracellular matrix regulation and fibrosis and things like that. Have you, have you thought about, or is there any value in exploring adventitial fibroblasts, which I imagine have kind of a similar contributory effect to the extracellular matrix remodeling?
Daniel, thank you for the question. Actually, that's something being investigated right now. So Cameron, student in our lab who's pursuing his master's thesis, is actually investigating if the adventitial fibroblasts are somewhat altered or not in response to the animal model of aortic aneurysm, and if the whole mitochondrial metabolism is involved in that or not. But to be honest, we don't have enough data to comment on that. But that remains a topic of investigation currently in our lab. Hopefully, we will have something soon on that.
Thank you.
Excellent. Thanks for those questions, guys. I'd like to see the, obviously through Dr. Fidak's lab, you guys have had some basic science training, and it's great to have you guys on the line listening and helping out with the discussion. Dr. Patel, really interesting stuff. I know the sections are keen to collaborate more. We're keen to get involved in these projects with you, and I hope the clinical spectrum becomes more aware of what the basic science groups are doing, because it's very interesting and it's very relevant. So great job to you. You're doing good work, and thank you for talking with us today. This basically concludes our aortic rounds for the calendar year. Thank you, everybody that's been attending throughout the year, and we'll reconvene with talks again in September. Okay. Thanks, Dr.
Thank you for the invitation, Scott.
Okay, guys. Bye.